Okay, so good day everyone. Okay, so my name is Halobadi. Okay, so due to the several reasons, so today class I will using the recording lah, rather than live class. Okay, so today we will finish up the nucleic chapter 1, which is we remain nucleic acid, another last biological molecules, and another one is analytical technique. Okay, analytical technique is one type of technique which is not a biological molecule, so we only remaining one biological molecule which is nucleic acid. Okay, so I think nucleic acid everyone should know, right? With the main component, the main example is DNA, RNA that you always heard about it. But what is DNA? What is RNA? So this is one type of the biological molecule. Okay, so nucleic acid is found. The founder of the nucleic acid is Friedrich Michel in 1869. So this is the scientist who first discover and isolate the component from the cells we call as nucleic acid. Nucleic acid. Yeah. And then why it is called as nucleic acid? Because nuclear, right? Nuclear is this component is found in the nucleus. We know that the cells containing nucleus, right? Whatever plant cell and animal cell, they containing nucleus. So when they isolated this component from the cell, they found that this component is found in the nucleus. So they found it, they named it as nucleic. And acid is because this property show acidic property, so they just put add on of it lah. So just acid. So the main name is nucleic acid. This is just a history for you guys to understand lah. But this one you don't need to worry about it. Will not come out in exam one. Yeah. The most important one is this. What is nucleic acid? So this is the basic structure of nucleic acid that you can see here. So this one is the I can say it is one type of polynucleotide already lah. So nucleic acid are the polymer that consists of nucleotide residue. So the basic monomer for this component is we call as nucleotide. Every biological molecule have their own uh, monomer, right? Except water. Water does not have monomer and polymer one. Like you learn about carbohydrate, the monomer is monosaccharide. And then you learn about lipid. The lipid also don't have the specific monomer. Like monomer, but you know that in triglyceride, it is forming from one glycerol and three fatty acid, right? And protein, they are also the monomer, right? Protein, the monomer of protein is amino acid. And then after that, we learn about nucleic acid. So the nucleic acid monomer is nucleotide. And it is located in nucleus of the cell only. We can only find nucleic acid in nucleus of the cell, except RNA. DNA all are found in nucleus, but some RNA, certain RNA and certain DNA, RNA can be found in cytoplasm. So not all the nucleic acid will be found in only found in nucleus. Just that the first scientist that found in this component is they isolate from the nucleus lah. Okay. But in real situation, after they discover not all the nucleic acid is found in the nucleus, but majority is found in the nucleus. Later we will see it. And the main component is carbon, hydrogen, oxygen, nitrogen, and phosphorus. And the main function is it is one type of genetic information. The main key function is that it consists about all of the genetic information. We know that DNA, RNA, all are the genetic, right? So nucleic acid is a monomer of them. So this is a basic structure. Okay. Okay. So as we know that we have two types of nucleic acid, DNA and RNA. So their full name is deoxyribonucleic acid and ribonucleic acid. You need to know the full name if the question asks about especially structure lah. Structure question, if they ask about the naming of the nucleic acid, you should write full name. You cannot write short form. Short form is only when you write in essay. Essay, yeah, you can allow to write short form. But in structure question, if the question is already specifically asking for naming, yeah, you need to write full name rather than short form. Okay. This one is the basic structure of the nucleic acid, which we call as nucleotide. So, as you saw there, as you saw here, yeah. So, nucleic acid containing three main components, three main subunits. One is phosphate group, PO4, PO4 2 negative at here, phosphate group. And another one is pentose sugar. This one, another one, pentose sugar, which we call as ribose, 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 ribose. Okay, another one is nitrogenous base. This one is, if you still remember in your SPA, you learn about, I think you also learn about, you can see the adenine, CGU, adenine, thymine, guanine, cytosine, and uracil, all these five, nitrogenous base. So this is the basic structure of the nucleic acid. So the bonding is all the phosphate group and nitrogenous base are bonded to the pentose sugar. So you can see that this one is carbon number 1, right? Carbon number 1, number 2, number 3, number 4, and number 5 at here. At here. Okay, so this one, the nitrogenous base is bonded with carbon number 1 of ribose, and another one phosphate group is bonded with carbon number 5 of the ribose. This one location you need to know it. Carbon number 1 and also carbon number 5. You need to know how to draw it O2. Yeah, maybe in examination they also ask you to draw. So this one is a full structure. So this one is a simple. If you draw in simple, it look like that. Just a pentagon at here, and then got one rectangle at there, and one circle at here. Just like that. Yeah. Yeah, this one is simple. When you draw about the new DNA, later we will learn about the DNA. When you draw about the DNA, yeah, you can write here. But if the question already mentioned, you need to draw about the monomer of nucleic acid. Yeah, you should draw this full structure, the full original structure. You cannot draw like this. If you draw like this, yeah, no marks. I can say. Okay. So pentose sugar. Yeah. So as I mentioned, nucleic acid containing two types. There are two types of nucleic acid, DNA and RNA. So why we have two types? The D and R are based on the types of the pentose, which is ribose sugar. So in ribose sugar, there are two types of ribose sugar. Either we call as deoxyribose and also ribose. So the ribose is that the difference is that in carbon number 2, the bottom part, ribose is hydroxyl group, is OH, with the carbon number 2. But deoxyribose, because of this deoxy, D means no, but absent. Yeah, right, absent. So meaning that the oxygen is absent. Deoxy meaning oxygen is absent, right? The oxygen is hilang, is disappear. So this one pentose sugar is deoxy, meaning that in carbon number 2, there are no oxygen atom. This one is just the normal carbon only. This is a key difference. You should know. You must know it. You must know it. So when you draw the monomer of nucleic acid, you need to carefully lah. One is got hydroxyl group. This one is RNA, ribose sugar. Well, we call as ribose sugar. That is why we call as ribonucleic acid, ribo. And this one is deoxyribonucleic acid, right? Why? Because deoxy. So they just take short form DNA, and then this take short form from RNA. So deoxy is there are no oxygen in carbon number 2. Okay. So yeah, this one is just a more larger part. I just want to show you carbon number 2 only. You need to remember carbon number 2, not other carbon, not carbon number 1, not carbon number 4, not carbon number 3. Carbon number 2. Yeah, you need to know lah. Okay. Next, we go to the phosphate group. Yeah. So later, just now we talk about the pentose sugar. So now we talk about the phosphate group. So whatever kind of nucleic acid, DNA, RNA, they have the same phosphate group. No difference. So the phosphate group is this. This is the PO4 to negative lah, the phosphate group the phosphate group uh chemical structure is PO4 2 negative, 2 negative. So this one is they will bond with carbon number 5 of the pentose sugar. So this is highly polar molecule, highly ionized oxygen atom, and provide the negatively charged to phosphate group. Yeah. Okay. Just okay. This one is very another important things lah. Nitrogenous base. So as I mentioned before, we have 5 type of nitrogenous base, right? Adenine, guanine, thymine, cytosine, and uracil. As you guys know that there are 2 main class of the nitrogenous base, pyrimidine and also pyruvine. Uh, Pyrimidine and also pyruvine. The difference are the structure of the ring structure of the base. Pyrimidine is more smaller. It only containing hexagon structure only. You will see here like thymine, cytosine, and uracil. All these are only neither uh single pentagon structure only. Just only single pentagon. Not pentagon, hexagon, sorry. Hexagon structure. Uh, and you see adenine and guanine is which is more larger. This is purine, which they containing 2 ring structure. One is hexagon and another one is pentagon. So they do look like that. So but in the end, you don't need to worry, you don't need to draw the original structure like this, don't need, you just know how to draw it like this, then it's okay. You need to know lah, pyramid is hexagon, pyramid is 1 pentagon and 1 hexagon. If you say that, or I want to draw, okay or not? Yeah, okay, but you need to make sure your drawing is totally correct lah. If your drawing is not correct, you will see that there are many double bond, right? Double bond, you also need to alert about it, and then the side product about it. Yeah, so not recommend you draw the full structure lah. Yeah. So this is the 5 nitrogenous base. So as you know that there are 5 nitrogenous base. So pyrimidine, there are 3 lah: thymine, cytosine, and uracil. And pyruvine, there are 2: adenine and guanine. So this one you need to remember which one is under pyruvine, which one is under pyrimidine. Yeah, so yeah, you need to know lah. About the polynucleotide. Okay, so the polymer of the nucleic acid, all of the biological molecule have their polymer. Like the carbohydrate, your polymer is polysaccharide. Like your protein, your polymer is polypeptide. In your lipid no polymer lah. But in nucleic acid, there are also their polymer, which we call as polynucleotide. So this is just a very long chain of the nucleotide. They join up together. When they join up together, they will form very long chain. We call as polynucleotide lah. So how they are formed? They are formed or same condensation reaction where the one water molecule is removed. Okay. So how they form? So the main form is you can see here. See here. This one. This one, if you see here, this is the one structure of nucleic acid, right? This one, you say G. Okay, UG, this is the basic unit of the nucleotide. And then one, you will see here, see this part, the phosphate group is bonded with the another pentose sugar, another type of nucleotide, which is C. The nitrogen number is C. We bonded with another nucleotide, we see. So this one is we call as phosphodiester bond here. This one, and also here. As also here. So the phosphodiester bond is formed between the phosphate group and also the pentose sugar, the ribose. Okay, so the forming the bond, phosphodiester bond is formed between the carbon number 3 at here. Carbon number 3. You see 1, 2, 3 at here. And you can see 1, 2, 3 at here. Phosphodiester bond is formed from carbon number 3. Between carbon number 3 of the ribose and to another type of nucleotide of phosphate group. Another phosphate group of another nucleotide. So this one is we call as phosphodiester bond. Yeah, you need to know the location before why you if you draw it, you cannot confuse it. You need to mention that the basic unit is carbon number 1 and carbon number 5. And if you want to draw the phosphodiester bond, it is located at carbon number 3 like that. Like that. Okay. So as I mentioned, two main class lah. So DNA and also RNA. So the main difference, I just show you the main difference. You can see DNA is double helix structure like that, meaning that there are two chains. One chain is at this side and another chain is at this side. Later I will explain more detail. But I just want to tell you that DNA containing two chains. There are two polynucleotide. Polynucleotide. So there are hydrogen bond between the nitrogenous base at here. But in RNA, there are only one polynucleotide only. Only one polychain, one change only. Only one change. There are no two chain. There are no two chain. And in nucleic acid, there are only either one or either two. There are no three chains and there are no four chains. Right? Two chains is DNA, one chain is RNA. Okay. So we go to DNA. So DNA, this is the one of the model we call as Watson and Crick model. Watson, Watson, and Crick. So these are two scientists. They propose the structure of the DNA. These are the two scientists that discovered the structure of DNA. Before that, we only know about nucleic acid. We only know about nucleic acid, but we don't know what are the structure of DNA and also RNA. But DNA is more important because DNA is carrying the genetic information, carrying all the genetic information. So DNA is more important compared to RNA in the research study. So as earlier, Watson and Crick they discover the structure of the DNA because as Watson and Crick model lah. So they found that the main point is DNA is double helix structure that consists of two polynucleotide chain coiled together. So this is like the helix structure that you see in secondary structure of protein. So similar, just that this is new DNA lah. Okay, so if you see the diagram, you will see the diagram is like that. This is DNA. This is DNA. But when we draw it, we will see like that lah. We will see like that, just like the 2D structure. This one is a real DNA that in our cells one. But this one is just a research purpose only. We just want to determine the nitrogenous base only. So if you see here, so this one is the full structure of the DNA. So when the question asks you to draw the DNA structure, first you need to know that DNA is double helix structure. Double helix meaning they containing two polynucleotide chains. So you need to draw two polynucleotide. And you need to know that the DNA chains, for each chain, they are anti-parallel to each other. Meaning that first is if there are the direction of the DNA for this one is 5 to 3. 5 pi to 3 pi. So what is 5 pi? What is 3 pi? 5 pi is we call as 5 pi end. 5 pi end. If there are the 5 at here, 5 pi end and also 3 pi end. Normally the left hand side is a 5 pi end and also 3 pi end. So this one 5 pi meaning that the ending of the DNA is at carbon number 5, and the ending of the DNA is at the carbon number 3 at here. Because phosphodiester bond is formed between carbon number 5 and also carbon number 3, right? Carbon number 3, and also the phosphate group that we locate at carbon number 5. So when we talk about the direction of the DNA, we know we use 5 pi end, meaning that the ending of the DNA 5 meaning it is ending at carbon number 5. If 3 at the bottom meaning that it is ending at carbon number 3. And then I need to tell you they are anti-parallel, meaning that if this 5 to 3 meaning here should be 3 to 5. So that is why that when you draw the DNA structure, you can see this is totally parallel. So we call as anti-parallel. They are anti-parallel to each other. Not parallel. If parallel meaning that If parallel meaning that they are in the same direction. 5 to 3, 5 to 3. No, they are anti-parallel, meaning that if this is 5 to 3 and this is 3 to 5. So you can see here, carbon number 3 is ending at here. So this one is carbon number 1, right? Carbon number 1, 2, 3. So the end at carbon number 3. So at here, you can see phosphate group is at here. So it is end at carbon number 5. So when you draw about the DNA structure, the full structure, you need to very be careful. Not just like draw like this, just draw the basic structure like this, the basic that you normally draw. You need to adjust the structure like this, become like this. You need to adjust. How you adjust like that? You need to draw lah. You need to make sure that the nitrogen base bond at here, this one is considered as number 1. So when you number 1, number 2, number 3. And you cannot like that, carbon number 1, number 2, number 3. Yeah. So number 1, number 2, number 3. Number 1, number 2, number 3. Yeah. So you need to alert it lah. Okay. Okay. So when you draw, when you combine it, and then this one is just that we need to know about the nitrogenous base. And finally, this is the whole structure, double helix structure we call as. So the DNA actually is look like that. When you observe under microscope, electron microscope, your DNA structure is like this. Not like this, not like this. This one we cannot observe. Even this one is atom, right? This is under the chemical atom already. For now, there are no such technology. You can see this kind of structure yet. Okay. Uh, Okay, so another thing I need to tell you is in DNA, they are linked together by phosphodiester bond lah. And repeating sequence phosphate deoxyribonucleic form the sugar phosphate backbone. So like this lah, I just like this lah. This one is we call as sugar phosphate backbone. Backbone. Like the backbone to the DNA lah. Okay, and then I need to tell you is the hydrogen bond between the nitrogenous base. Yeah, in nitrogenous base, the as I mentioned, we have 5 type of nitrogenous base, right? Adenine, thymine, guanine, cytosine, and another one you will see. So for DNA, there are only 4 types of nitrogenous base. Adenine, thymine, guanine, and cytosine only. How about uracil? Uracil will be specified for the RNA. So one of the key characteristics to differentiate DNA and RNA is to determine the nitrogenous base. If we found that the nitrogenous base only containing thymine, the most important is thymine. If containing thymine, then this strain is DNA. If we containing uracil rather than thymine, meaning this is the RNA. So thymine and uracil, they can change one. Thymine is specified for DNA. Uracil is specified for the RNA. Both of them will be bonded with adenine. And cytosine and guanine, both DNA, RNA are same. Both of them containing. Just that either thymine for DNA or uracil for RNA. So the most important thing is the number of hydrogen bond. Adenine and thymine or adenine and uracil, they are bonded with 2 hydrogen bond only. 2 hydrogen bond. You need to know number, but for cytosine and guanine, there are 3 hydrogen bond. I just want to tell you, there are 3 hydrogen bond. So I repeat again, adenine to thymine or adenine to uracil, they are only bonded with 2 hydrogen bond. For cytosine and guanine, they are bonded with 3 hydrogen bond. So how to memorize it? AT. So double bond, double. CG. So 3 double bond, 3 hydrogen bond. So ATCG or AUCG. ATCG, you just memorize like that. ATCG, AUCG. AT is for DNA. AU is for RNA. You need to know that. Okay, so that's all about the DNA. So very simple, right? Not too hard lah. So and then this one just that I will tell you that last year, your senior, last year just come out as a question about want to explain the DNA structure. Yeah. So another one maybe this year come out RNA. RNA never come out yet in the real examination. Yeah. So RNA full name is ribonucleic acid. So as I mentioned before, thymine is replaced by two uracil. So in RNA there are no thymine. Will be replaced to uracil, but it will still bonded with adenine. Still bonded with adenine with two hydrogen bond. And cytosine and guanine just bonded with normal three hydrogen bond. Say it only consists of single polynucleotide. So like that, just like that. Only see one change only. Compared to DNA, there are two change. You can see here there are two change. One change is like this, and another one is like that. Okay. In RNA is very special. DNA only containing one type. We only containing one type of DNA. There are no such thing as we have two types, three types of DNA. Everyone have the same structure of DNA. Everyone. Just that the sequence are different The nitrogen base are different. The order sequence for every person is totally different For RNA, there are three types of RNA. First one is we call as messenger RNA. Another one ribosomal RNA. And final one is transfer RNA. We call as tRNA, mRNA, and also rRNA. So both of them, three of them have their different function. So we go to messenger RNA first. Okay, so this one before that, I just want to tell you more easy to tell you the function more easy You need to know that DNA is inside in the nucleus, but mRNA is not inside your nucleus one. It does not contain your nucleus. Not sorry. mRNA can be inside your nucleus and also from the at your cytoplasm. So mRNA is synthesized from DNA one. This is just a short change only. Short change. You can say it is like a copy segment from DNA, but very short only. Very short. Just a copy. We will process we call as transcription process. So in DNA, we need to transcribe into mRNA. You can refer like transcription process is just a copying from the DNA only, but only short segment. Not all the containing all the sequence in the DNA will be copied. No, only certain segment only. This one we call as transcription process. So mRNA only 3 to 5% of the total cell and consists of single stranded as I mentioned and containing complementary codon formed by a transcription of a single gene on a single DNA strand. So this one is that I just tell you as I mentioned just now This is like the copying from the DNA, which complementary, meaning that what you see in the DNA, they just copy. Like you see, like they will choose either which chain as a parental chain. So maybe here, they choose this as a parental chain. So they will form another chain we call as mRNA like this another chain mRNA like the left hand side. So C G. So this is G, but in RNA no T. So we will replace to U. G U G C U G A A. So this one, the left hand side will be another strain we call as mRNA Yeah, mRNA like this like this So it carry the genetic code from the DNA in the nucleus, and then later this mRNA will go to the cytoplasm to synthesize the protein through the translation process. So actually this one is there you will learn about the more detail the mechanism in SEM 3. So for now, I don't want to make you pening kepala. You just know that you just want to know that mRNA is synthesized from DNA. How they synthesize? They use one of the stranded from DNA, any strand. Either they want to use 5 to 3 or they want to use 3 to 5, but normally is they use 5 to 3 lah. 5 to 3 is the most they use. After that, they will do the mechanism we call as transcription process to form mRNA. So what are the function of mRNA? Why we want to form mRNA? The key function is to synthesize protein. We only the mRNA now they were coding all the sequence that can produce the protein by. So this coding we will call as genetic code. So different genetic code they will code it code with the different type of the amino acid. So you will see this all color, one color are one amino acid. So they will code with different amino acid. So this one, you will learn more detail in Sem 3. Very detailed. Later you will learn more detail. For now, you don't need to know this. You just want to know that mRNA from DNA. And then this mRNA is containing the code, genetic code, and which we can code with different type of amino acid to produce protein. Just that all. You just want to know this kind of function only. And another one, RNA, ribosomal RNA. So as you see this ribosome. So ribosome, right? Ribosome meaning that this RNA is located inside the ribosome. So this RNA is totally at the cytoplasm. Right? mRNA, they synthesize in the nucleus, and after that they will move out, they will travel out from nucleus into cytoplasm. But this one is totally inside the cytoplasm because they locate in the ribosome. Ribosome inside the cytoplasm, but they locate in the ribosome. So this one is to aggregate with protein to form the large sub-ribosomal unit and small ribosomal unit. So this one is just to form the structure of ribosome lah. You can say that this is like the key structure. Key structure they combine with the protein molecule to form the ribosome. So this also the primary function is to protein synthesis. And the most abundant form of the RNA in living cell lah. And the sequence of the RNA have been used to characterize the evolutionary relationship between organisms. And finally consists of a single polynucleotide chain synthesized in the nucleus. So this one is that more to bioinformatics lah. Bioinformatics lah. Just that we need to know about the main important is RNA is one of the parameter that we can determine the species, especially in bacteria. In bacteria, we can directly using the RNA we call as cysteine S lah. Cysteine S and RNA directly we will know that which kind of bacteria is it. The naming of the bacteria. But yeah, this one you don't need to know very much lah. Just that one of the function of the RNA. And finally, tRNA, transfer RNA. So this one is the structure is totally different from the previous one. The previous one is just a single stranded. Yeah, they single stranded, but they are just a one line only, one chain only, one line only. But you see the tRNA, the structure is like this. They have the loop structure and 3D structure. They are single stranded folded into a 3D clover leaf shape. This is the spatial shape in tRNA. Sorry. tRNA we call as clover leaf shape and held by hydrogen bond. So tRNA they look like double stranded, but they look by the double. They containing the double chain. There are two chains, but they are only single chain only. Just that they can form this single chain. They can form 3D structure while they hold it by hydrogen bond. You can see here. There are hydrogen bond here. You can see here hydrogen bond. So the hydrogen bond is totally same same in DNA. So CG containing 3 hydrogen bond, AU 2 hydrogen bonds. Totally same. So they can form the loop clover leaf shape. So you can see they have the loop at here. They have the loop at here. So in tRNA, they are also 5' to 3' lah. So 5' end and 3' end. 3 pi n. So 5 pi n is will be start, and then after that, the 3 pi n will always end in the base of sequence of CCA and act as the amino acid binding site. So you see here, this one, 3 pi n is the place to bind with the amino acid. Yeah. So this DIA, I give you the function is that it is the key function to synthesize protein. They will use the information from the mRNA to as the help to synthesize protein. Yeah, so the rtRNA, you will want to synthesize protein, you still remember you want to synthesize protein, you need amino acid. Right? You need the amino acid. So amino acid cannot bind together one. In our cell, they just not just that or simply lah. Or awak kawan like glycine and glycine aspartic acid kami dua kawan, so kami join together form dipeptide polypeptide. Cannot, no such thing. The joining is based on the sequence of the mRNA. The joining is based on the sequence of mRNA. This DNA is transfer amino acid to the ribosome. Already say lah, transfer. It look like the macam lorry lah. You can say that DNA is like a lorry. Dia ambil amino acid. So kepada transfer to the mRNA to see ini betul ke tak. Oh, ini betul. Okay, saya join. Ini tak betul. Oh, tak apa lah. Tak apa like method like the glycine lah. Glycine. Okay. See, this mRNA got the coding for glycine or not? Oh no. So this amino acid will not join at here. So they will see another methionine. Oh, got methionine. Okay, they will join. Like that. Oh, got aspartic acid. Oh, they will join. So yeah. So this is just like you can say that DNA is like a lorry lah, like a car transport transfer, right? So they will bond with amino acid, all the amino acid, 20 types of amino acid. So in our human, there should be at least 20 types of DNA. So how they join the amino acid is that they simply bond it, simply carry the amino acid. No, they carry the amino acid based on the we call as anticodon of the DNA at here. Anticodon of base triplet sequence and is directly related to the sequencing amino acid carried by the DNA. So this one is the key parameter to let the amino acid to join. Like methionine. What methionine? Maybe like CGG is methionine. For example CGG is glycine. Okay, so only glycine can join this. Other type of amino acid like aspartic acid, tryptophan, or other glycine cannot join. The lysine cannot join. All that cannot join. Only the methionine can join only. For example, there are the specific sequence but I not memorize it. I just give the example So CGG maybe is methionine. So only methionine can join at here. They can only the methionine can carry by this DNA. Other type of you want to join glycine, they have to other type of amino acid. So you see here this mRNA. This mRNA, right? So they will carry the amino acid, they will carry the amino acid first at surrounding and in the cytoplasm first. And then after that, once they carry, they will go to mRNA to check whether they are corresponding to it or not. They are complementary to or not. If they are complementary, we call as codon. So now codon, mRNA is codon. If complementary to mRNA, then amino acid will be released to join to form the protein. If not, complementary to each other, then this amino acid will not be released and this DNA will directly come out. They will go out and another which is complementary DNA will join together. So these are DNA. So the DNA, you can say that the function is like a transport that carry the amino acid and then go to the mRNA to check whether this mRNA is mixed with matched with the tRNA or not. If matched, then this amino acid will be released and go to form the protein. And if not, this amino acid will not release, so it will this all the DNA molecule will directly coming out and allow another type of DNA lah. So like that lah. Okay. So this one, the detailed mechanism we call as translation process. So the translation process you will more detailed learn in semester 3. So actually this one is quite difficult. I can say maybe you are not very understand. I hope my explanation can give you understanding lah. I already try my best. Because this one is more detailed when you learn in sem 3. But unfortunately, you are not in sem 3 yet, so your syllabus also not learn about the transcription and translation. But the function of the tRNA is actually is we call as translation lah. So when you want to write about the function of tRNA, mRNA, RNA, actually you need to write about the transcription and translation process. We call as central dogma theorem lah. But due to you are in sem 1, so you can only write the general function as I say before. Okay. So now we go to difference between DNA and RNA. I think this one I already mentioned many times lah. So I think I need to tell you about the location lah. So as I mentioned, DNA normally is located in the nucleus. Majority is located in the nucleus, but some of them will be located in mitochondria. Mitochondria and also chloroplast. Chloroplast and mitochondria, they may containing DNA one. May containing, but majority of the DNA, all the DNA that carries genetic information is inside your nucleus. And RNA can be found in anywhere. Cytoplasm, nucleus, ribosome, all can found. Cytoplasm, yeah, mRNA, RNA, tRNA, both three of them can be found. Nucleus is only mRNA. mRNA, normally mRNA, because they synthesize it. Ribosome is rRNA. And definition, I think I don't need to talk about that lah. So the most important, I just want to tell you the nitrogenous base lah. So RNA, UAC, DNA, thymine, and sugar position, it has 2 deoxyribose, meaning carbon number 2 lah. Carbon number 2 does not have oxygen. So that is why we call as deoxyribose. Deoxyribose DNA. And it has ribose, meaning that it is normal lah. They are containing the carbon number 2 is hydroxyl group. They're containing oxygen. And the function, normally the DNA function is to as the genetic information. They carry genetic information, what? DNA. But RNA don't carry genetic information. They're just a transmission of the genetic code. They are necessary for the protein creation, protein synthesis. Okay. And predominant structure, so DNA normally is double-stranded. All of the DNA are double-stranded. RNA is single-stranded. And the distance, the number of the nucleotide, DNA is more longer, and RNA is more shorter. And propagation, DNA replicate on its own, meaning that this is a replication process. This replication process. Oh, sorry, this one should be RNA. Sorry. Okay, DNA can be replicated, yeah, based on their own mechanism. RNA cannot replicate on their own. They need to realize on DNA one. They realize on DNA. So mRNA, like, will not say that mRNA, if they are already got one mRNA and then this mRNA can copy to another mRNA. No, cannot. They cannot copy to each other one. The mRNA only can be synthesized based on the DNA. If they want to produce the mRNA, they need to according to DNA one. They cannot like DNA. DNA can directly copy one. They can copy and paste. I say it's easy. DNA can copy and paste. But mRNA or whatever RNA, they cannot copy and paste. All of them must rely on DNA. Ultraviolet damage, yeah, DNA is more susceptible to UV damage, UV damage. But compared to if RNA, more resistant. Okay, so that's all for the nucleic acid part. So now we will proceed to the last topic, which is the analytical technique. Okay. So based on the syllabus, analytical technique, actually this one is the general topic only lah. Analytical technique. Analytical technique is the what we say that this is like the want to know the component of the component of the like DNA like that lah. But in your syllabus, you only learn about two technique only, two method only. The first one is paper chromatography, the first. And another one we call as gel electrophoresis. Okay, so what are they? What are they? Totally new to you guys, right? So this topic is new, that you never learned about in SPM. So this is totally new. So paper chromatography and also gel electrophoresis. So this is just a two example only. Actually, there are many type of analytical technique also. If we think chromatography, there are many type of chromatography. But in your syllabus, you only learn about paper chromatography. So this paper chromatography is one type of the experiment, okay, assignment lah. Yung kerja khusus. One of the experiment that you need to conduct in your school lah. I don't know your school already start or not, but just that you know lah. Paper chromatography is one type of the experiment. I think it's the first one lah. First or second lah, based on your school lah. So you need to know the definition of the chromatography. So chromatography, actually this is a carbon lah. Chroma and graphy, based on the Latin word, chroma is color, color. So graphy is white. So they just chromatography. So the main function is to describe the separation technique based on the between the mobile phase and also stationary phase. So in simple, chromatography is the separation technique. If you know that in one mixture, I say more in one mixture, one chemical mixture, they containing many compound, many component. What they may be containing salt, they may be all those salt. There are many type of salt. So not only salt lah. They containing salt, they containing organic A, organic B, organic C. So there are many different organic compound in your chemical mixture. So chromatography technique is to make sure that allow you to separate the content of the mixture so that you will know, oh, this chemical containing which part of mixture. When if you don't know, unknown, we call example unknown, so that you know, or you run the chromatography, or then you know, or this solution containing organic A, containing organic B, or containing organic C, like that lah. So this one is the basic principle of the chromatography lah. But in you so why we want to use chromatography? So you need to separate two or more compound in a mixture based on the different in property. So they is based on the different property. Either based on polarity, solubility, ionic strength, or size. Size. Yeah. So in chromatography and paper chromatography, they are containing two phase we call a stationary phase and also mobile phase. Yeah. So stationary phase is the place where your separation occur. Separation occur. Meaning that when your paper, you see this paper, uh, like see you this. This one we call as your paper. This one is paper chromatography lah. Paper chromatography. You look at a paper, the spatial paper, not like the A4 paper that you use in your book. This is a spatial paper. Paper. They are the spatial paper. So your paper will be your stationary phase. Why? Because your paper is the place that your separation will be occur. So separation will be occur. So what is how about mobile phase? So mobile phase mobily is liquid fluid lah that flow through over the stationary phase to carry the sample. The mobile phase like the transport process to transport the sample into the stationary phase. Because stationary phase is solid. Itu paper right? Paper. You can you imagine if you directly throw the sample into it, can you draw a separation? Cannot. No, nothing. So you need to have a pushing process, the transport process. to transport the sample. So normally you see here, see this diagram, this is paper lah. So this one is all the paper is we call as stationary phase, and they will start at here, meaning that you will dot your sample, your solution, you not directly, not directly like this, just immerse your solution like this. No, this solvent is we call as solvent is mobile phase one. So any solvent lah, solvent. So normally solvent we use will be the or polar molecule lah. Okay. Polar like hexane, yeah, hexane lah. One of the molecule is hexane. Okay. So anyway, the most important I need to tell you about the theory is the origin at here, meaning that you dot the sample, and the sample is not direct. You directly throw it away, directly pour it away. No, you awak tak boleh tuang the dekat sini. Tak, they just dot only. They just dot few dots only at here, few dots only. So based on your sample lah, if your chromatography paper is very big, you can drop many samples. So normally in your school, I think your chromatography is they already cut for you lah. Not will not be very big one, not like that lah. This one is quite big lah. This one is quite big one. In your school, I think they already cut down. They will ask you to cut down to into more smaller like that lah. So you will only dot one sample here. So you take your sample is solution, right? Take your unknown sample, dot at here. So meaning that the sample was starting to move from here upward. So how to move? If let's say if you does not have mobile phase, does not have any solvent, can your sample move? Cannot. Your sample is also solution. They also need to have a water, something like that. Water, right? You kalau awak nak sapu lantai, awak nak mop lantai, awak nak cuci lantai, boleh ke tak ada air? Tak boleh kan? Memang ada air. So that you can remove, awak boleh cepatkan semua itu daun-daun atau sampah-sampah keluar kan? Compare to no air. Kalau tak ada air, awak just guna penyapu je lah. Suka, right? So fair. So yeah. So mobile phase is the place. So after you already put the sample, dot your sample, so you will immerse this paper into the solvent. So once they immerse into the solvent, so your solvent was starting to immerse, they will flow lah. Your solvent will be uh uh flow through over the stationary phase. So they will flow until end lah, until end solver fund. So at the same time, your chlorophyll not chlorophyll, your sample, your unknown was starting to separate lah, based on the here. So when you see this diagram, meaning that this sample containing three pigment, or we call as three pigment lah, or we call as containing three different mixture lah, three different things. So red color, purple color, and blue color. And then you need to know how to calculate RF. So this is just a parameter to know that the distance, to know that the separation is good or not lah. Whether the separation is good or not. So the formula is distance of component divided by distance of solvent fund. So meaning that we will see this example. So your diameter distance of solvent fund, you see here. So from origin until the end is 10 cm. So your diameter is 10 cm. And this component is based on the different pigment. If red color is 2 cm. So 2 over 10 is 0.2 lah. So your RF for red pigment is 0.2. So if for your purple is 6 cm. So 6 over 10, your RF value is 0.6. And for your blue is 8 cm, right? So 8 over 10, your RF pigment for blue color is 0.8. So very easy. So normally this one, if they come out, normally they will come out objective lah. So they come out objective, wow, bonus mark I can say. Very easy. Completely very easy. I can say 99.99999% is you can score it. It's very easy. You just know the formula, then you will directly know. Yeah. So this is the answer lah. Okay. Okay, so that's all about the paper chromatography. Now I will proceed to the gel electrophoresis system. Okay, so this one is another technique, one of the famous technique that you need to know too, and based on the syllabus. So this is the technique to separate the DNA molecule and also protein molecule based on their size. We know that DNA is very large, right? And also protein. Protein and DNA, both of them are very large molecule. So we need to, in the research purpose, we need to separate their size. We need to do the separation according to the size of the DNA and protein lah, for the research purpose. So in your STPM, sorry, there are no experiment about this. You have no chance to do the experiment like that. But in university, yeah, you will able to do. Especially if you go to genetic profile, genetic, or you go to biochemistry or biomedical, biotechnology, yeah, you will always see this stuff. So this concept is that there are three main functions lah. It is a laboratory technique to separate DNA, nucleic acid, RNA, protein molecule based on their size and also electrical charge. Okay, electrical charge. Okay, so general step in electrophoresis, I just tell you lah. If you want to run a electrophoresis, so firstly you need to prepare a gel. So like agarose gel, why we call as agarose gel? Because one of the gel we know as agarose. Agarose is one type of the carbohydrate, the polysaccharide that you never learn it. This is because this is what you say multi heteropolysaccharide. They are containing many different uh, carbohydrate monomer. Not like the that you learn is homopolysaccharide. This one is heteropolysaccharide. So you don't need to know the structure. You just want to know that one type of the gel we call as agarose. So this one is the process. Firstly, you need to prepare the gel first. You need to prepare this first. This solution first. Agarose gel. Prepare gel. Of course, at the same time, you need to prepare sample. So you need to got the sample first. If you know you don't have the sample, how do we done? Right. So then after that, when you prepare all this, you need to set up the electrophoresis chamber like that. Like that. Like that. Okay. Like that. So after that, you see that. So there are the valve at here. There are the valve at here. So this one, they will have the loop hole at here. Hole at here. So this hole is the place where your sample is locked into here. You put your sample into here. So you load, we call as load lah, not put lah, load the sample. Load the sample into the valve. So once you load the sample, your sample is already inside the valve, already inside the hole, or that you can run your sample, and then you will see the image like this. You can see the image like this like this. So this image is based on the separation of the DNA, RNA, or protein molecule based on their size and also electrical charge. Okay, so yeah, there I just share to you lah. This is experiment, the result that I do in my university. So look like that lah. But actually this one is fail one lah. Not say fail lah. Not very good result lah. If this kind of result we need to redo lah. This one is more clear lah. You can see here more clear. You need to see the result like that. So this one is more clear lah. But for this one is fail lah. Okay, you consider fail. You need to redo lah. Okay. Like that. More clear lah. This one is pass lah. So you can see that DNA ladder is like the ladder, like a ruler. You can say it like the ruler to determine the you know the size of the DNA or DNA lah. Directly know lah. Because if you does not have the DNA ladder, can you imagine you like just like that only? You know the DNA ladder, you just like that. Can you know lah? This side is ini accurate macam-macam ini berapa size tak tahu. So you need to have the DNA ladder. So DNA ladder will provide you so that you already know lah. This side is 100, 1,500. This one is 700. So you can know that sample A is 700. And sample B, there are 3 bands, right? You can see there are 3 bands. So there are 3 different sizes. There are 3, 700, 500, and 200. And you can see here. See here. So that's all. And then I need to tell you that their electroporation is separated according to the size of the DNA, meaning that the more smaller size of DNA will more faster. You will see here 100, 1500, and this one is 200, right? The more far away from the hole, the more smaller, and they will go more faster one, very, very fast one. But for the larger side, they will go very slow. So that is why that they cannot move too fast one. They very slow one. Based on their size, yeah. And also the charge. This one is negatively charged. They will move toward the negatively charged. So after affect the electrolysis, your sample size is more will be affected lah. Smaller will be moved very fast lah. And your gel concentration also can be affect, and your voltage also can be affect. But normally it's a simple size lah. Gel concentration and voltage normally fixed already. We do the experiment, we already fixed lah. But sample size is we cannot fix lah. This one is very you need to know lah. And there are many types of electroporation, not only agar gel. Agar gel normally is for DNA only. And for protein, we have another electroporation we call as SDS page. But the concept is totally same. What you apply in agar gel, they are same. Just that there is quite something different lah. Yeah. And another one is paper electroporation like that. So this one is protein, we call as isoelectric focusing. So this one is based on the pH value of the protein. Protein. Meaning that normally you load the sample at the middle. At the middle. And then after that, you run the electrolysis, the protein will migrate to the isoelectric pH. So that where they have no net charge, meaning that not all the protein is neutral in pH equal to 7. You should still remember that some protein is acidic, some protein is basic, some protein is polar, some protein is non-polar. So especially in acidic and basic amino acid, their isolated electric point. For acidic amino acid, their isolated electric point, meaning for no net charge for the this kind of amino acid will be at the lower pH one. They will have the lower pH one. So they will move at here. For basic, they will move at here. So like that lah. And this one is SDS page. So this one is just based on the protein size. Just now this one is based on the charge, based on the isoelectric. This one is SDS page, which is according to the size of the protein. So different protein have different size, right? So this according to the size of the protein lah. Okay, so I think that's all for class. Okay, so as I mentioned, starting today class, I think I need to using the recording lah. So I will change the date of the class time, and I will post it to the Telegram. I will make an announcement, so there are no more live class. So I need to sorry about that because I'm very busy on my university, so I cannot have my time is not flexible lah. Maybe sometimes I'm free, sometimes I'm busy. So yeah, so I think that's all for today. So I hope that we will enter chapter 2 starting next week lah. So the starting chapter 2, we were using recording lah. So I will share the time that I will release the video. So I think that's all for today. So if you have any question, you can ask in the group one. You don't want to ask in YouTube lah, because YouTube I cannot see the comment. Very bad to open the comment from YouTube. You directly ask in the group. You do not understand, you directly ask. Okay, so I think that's all. So thank you for your watching. So hopefully this chapter you guys can understand lah. So okay, so thank you and goodbye. Have a nice day. Bye.